Hello. Do you hear everybody? Me?
Good morning.
Good morning.
Good morning İbrahim Hocam.
İyiyim Nazım.
Good morning ladies and gentlemen. Teşekkürler hocam. İlahiyle konuşacağız. Now today we are opening the second day oral presentation session. This is the first session. The chairs are Professor Dr. Sevgi Kolaylı and also I appoint Ibrahim Türkmen, chairs of this session. Sevgi Hocam. Now I want to open the first session with the normally we change the presentation. OP1 will start normally Ahmed Ali India, but I think he cannot enter now. He will come. And the second presentation, Sevgi Hocam hazır mısınız?
Evet, hazırım.
Hocam,
I'm ready.
O zaman siz hem oturum başkanısınız hem ilk sunumu size vermiş olalım. Yes, Professor Dr. Sevgi Kolaylı from Turkey will start the first presentation, anti-diabetic activity of commercial ethanolic propolis extract from Anatolia. Please share screen your presentation. I would like to inform that we will make all the presentations in order, and at the end of the session we will have discussion. Hocam ekranı paylaşır mısınız?
I want to participate, but I need time.
Hocam sunumunuzu açın kendi bilgisayarınızda.
Sunumum açık ama direkt olabiliyor mu?
Hocam yani, alttan share screen diye bir yeşil bir şey var.
Tamam. Bakalım olacak mı?
Onu tıklayın. Ondan sonra bir saniye.
Olmuyor.
Evet, şu anda, tamam şu anda olur hocam.
Disable.
Tamam şu anda tekrar yapın.
Tamam hocam.
Hocam screen'den şey yap. Çok.
Evet.
Böyle.
Tamam.
Hocam tam ekran yapalım.
Tam ekran. Okay.
Okay. You can start. You have 10 minutes.
Okay.
Please summary, only summary, not more information, not general information, your results, your aim and results.
Thank you. Okay. In this study, we tried anti-diabetic activity of commercial ethanolic propolis extract from Anatolia. Diabetes is very important disease in the time, and we have a lot of 3 or 4 types, especially type 2 non-insulin dependent diabetes mellitus is very big problem, and the type 2 diabetes drugs are mostly amylase and glucosidase inhibitors. Today some competitive inhibitors used the treatment of the disease. For example, acarbaz, miglitol, voglipose as used as anti-diabetic drugs, and some natural inhibitors are still investigating, and some herbals and propolis have anti-diabetic activity also. There are some studies in the literature. For example, Malaysian propolis compared with metformin and have good potential anti-diabetic activities. Another study again, propolis extracts have good potential antidiabetic activities by inhibited glucose 6 phosphatase enzymes was showed. And another study again, chlorogenic acids and rutin, they are two important polyphenols found propolis almost, and have good antidiabetic activity potentials. For this reason, we tried propolis for antidiabetic activities. And propolis is a bee products, very important bee products, and collected from hives by raw propolis. But we use it, especially ethanolic propolis extracts, in some apitherapy applications. And propolis searches almostly trees, barks, and Turkey is a very rich about propolis searches. And propolis is getting increasing propolis used by people, and especially antioxidant, antimicrobial, anti-inflammatory, anti-tumoral agent, antiviral, antidepressant, and hepatoprotective and antidiabetic activities is very important for propolis used. And propolis, two important parts have, and waxy and balsamic. We interesting especially balsamic parts. And the balsamic parts have many different polyphenols. The polyphenols is different, species differ from the region, from propolis searches. And Turkey is very rich about propolis searches. And in the study, we tried in vitro and in vivo antidiabetic activity, especially commercial propolis, not prepared from raw propolis. We supplied commercial propolis and ethanol propolis, and the propolis obtained from a big manufactured firm from Istanbul, B&U named also. And we analyzed, this study was carried out in 3 different stages. Firstly, propolis drop analysis. The second, in vitro and diabetic activity, and third, in vivo diabetic activity, animal studies also. Propolis extract, the propolis extracts analyzes pH, total phenolic contents, total flavonoid, antioxidant capacity, and phenolic composition analyzed also. And the results, and two enzymes, amylase and glucosidase inhibition also tested a spectrophotometric assay. And animal study was carried out in Algeria, our collaborative study, Professor Dr. Nurettin Decbli, and 25 male Wistar starts rats was used at ethical committee report used. And firstly, we tried acute toxicity tests for the propolis, and we found that no acute toxicity, all the rats was not showed. And induction of experimental diabetes, diabetes in the rats was getting streptozotocin injection, and 65 milligram per kilogram was applied after overnight fasting. And first 2 days after all rats was showed anti-diabetes. And this is our experimental protocol. First group, control group. Second group, only diabetic group. Third group and fourth group, propolis applying groups. And the results and propolis concentrations and composition and phenolic composition of propolis, and we found the propolis have a high amount of caffeic acid phenol esters, hesperetin, creatine, pinosempirin. They are a very important flavonoid also. And enzyme inhibitions, two enzyme inhibition we worked, and our propolis showed a good inhibitor effects, both of the enzymes, and the inhibition compared also acarbaz drugs. And we also used water consumption of the rats, and the consumptions was related anti-diabetic diabetes, and glycemic index was also measured by glucometer every day, and the results showed that our propolis samples, especially concentrated samples, showed a good inhibitors. The glycemic index was getting decreased. And another parts of the study was histopathological study, and pancreatic tissue was investigated, and the results showed that our propolis extract showed a good effect on Langerhans islets, and the study results showed that first I want to say this is the preliminary study, and the study we planned a big study again, the propolis samples, and but the propolis, Anatolian propolis extracts have a good potential anti-diabetic activities, and no acute toxicity was showed, but we need further studies in phase 1, phase 2, and phase 3 studies also. And thank you very much for your nice organization, although pandemic. And thank you. And again, Nurettin Dejli, our collaborative study in Algeria, and Hüseyin Şahin, my PhD students in now he is in Giresun University. And thank you again, B&U Propolis Manufacturing firm in Istanbul, and supplied propolis samples. Okay.
Okay, thank you, Professor Sevgi.
Kolaylı.
Kolaylı. And you are also chair of the session with Professor Dr. Ibrahim Tümen. I will give my order to you, and you can announce another presentation. Now I will share with you the new program, and this is the Ahmet Ali, the second presentation. You can introduce. İbrahim Hocam. Sesinizi açın. İbrahim Hocam. Sevgi Hocam siz sunum...
Duyuyor.
Duyuyor musunuz?
Okay.
Tamam.
Duyuyorum
Ne?
ben.
Okay.
Okay. Thank
İbrahim
you,
Hocam.
Dr. Şekeroğlu. First of all, I would like to thanks to Dr. Şekeroğlu and his team. Special thanks to for kindly and nice symposium. This symposium is very important. This MESMAP. Today is we will start sixth symposium. I think. Is it true? I think.
Yes.
Yeah. Okay. Again, good morning. Now, this presentation belongs to Mr. Malik and Mr. Ahmed Ali about preventive of advanced and glycation products by the seed extracts of Nigella sativa. Please, Mr. Ali.
Ahmed Ali.
Please,
Yes,
Mr. Ahmed Ali.
A very good morning to all of you. And thank you, Professor Dasim and your team, Dr. Sevji, the secretary of this conference. It's an honor for me to be a part of this MESPAP. It's third year. I've been attending. I started my first presentation and attending this conference in 2018. That was MESMAP-4. We were looking forward, but nature has its own. So anyways, I wish all the best to Professor Nazim and all the members of organizing committee. And last but not the least, I'm thankful to Professor Nazim for giving me this opportunity to present my work. There is slight spelling mistake in the title of this. The title of
Can
this my
share.
presentation
You
is.
have presentations.
Yeah, I will share.
Please share.
So, is it visible now, Professor? Okay, is my presentation visible? Yes, thank you, Professor. Okay, so the title of my presentation is Prevention of Advanced Glycation End Products by the Seed Extracts of Nigella sativa. The common name of this seed in English is black cumin, and it is a very well-known herbal medicine in Arabs and all over the world. They have been used for different purposes, but my focus is on the biochemical events which happen during the diabetic process. So just a brief about, because we are all concerned about this diabetes event. It has become a major metabolic disorder or non-communicable disease, and almost by another 20 years or 30 years, 50% of the population will be residing in developing and developed countries. So basically it involves the impairment of glucose metabolism. I'll not go into detail of this, what happens during the pathophysiology of this disease, but the modern theory of diabetes suggests that it is an implication of oxidative stress and glycation. And because of these modifications in the cellular systems, there are many secondary complications which are associated with diabetes. So glycation is a non-enzymatic process which happens between the sugar and any other molecule which has a amino group. Okay. So this is a non-enzymatic reaction. To differentiate it from a enzymatic equivalent, that is glycosylation in the endoplasmic reticulum and Golgi apparatus. So there are several stages before this reaction is complete: early, intermediate, and late stage. At the end of the reaction, a group of molecules are produced, which are known as advanced glycation end products, which have been implicated in many diseases because of their capability of changing the structure of biomolecules. So you can see the scheme here. Sugar and proteins react, and in the initial stage, a SIF base and amadori bases are produced, which further undergo modification and dicarbonyl compounds are produced. And finally, you see advanced glycation products which alter the macromolecules. So we can see that hyperglycemia leads to an increase in the ages, and these increases alter the molecular structure. There is another pathway by which ages work. They bind to their receptors on the various cells, and they increase the expression of nuclear kappa B, nuclear transcription factors like NF kappa B, ultimately leading to an increased oxidative stress, and finally causing a group of disorders like Alzheimer's and nephropathic and retinopathic and cardiovascular diseases. So people have been looking for this inhibitors for this age, and some synthetic compounds have been synthesized, and some natural compounds are being looked for. So these inhibitors can interact or can inhibit the interaction between the sugars and proteins, or they can inhibit the formation of amadori products, and they can inhibit at later stages. So among the natural compounds, plants and among the natural inhibitors, plant extracts and formulations are being studied. This is the cellular mechanism by which toxicity of glycation is suppressed in the systems. We have enzymatic systems like aldehyde reductase, glyoxylase, amadoriases. We have protein degrading systems, vitamins, uh, synthetic compounds like aminoguanidines, and world has been looking for anti-glycating agents and antioxidants from. So my focus is to characterize the role of Nigella sativa seeds in the process of glycation. We all know that these seeds are quite rich in carbohydrates, proteins, amino acids, and volatile and fixed oils. One of the major phytoconstituent of this uh, plant is, or this the seeds of this plant is thymoquinone, which possesses various activities like antimicrobial, anti-tumor, antiviral, anti-inflammatory, anti-diabetic, and so on. So the structure, these are the four, some of the major phytoconstituents. As I mentioned, that one of the major constituent of this is thymoquinone. We have recently produced, I have recently published a review article on this, where I have mentioned in detail about the properties of thymoquinone and Nigella sativa. So what we have done, we have collected the seeds, and then we have prepared the plant extracts, maybe aqueous or methanolic, depending on our requirement. Then we did antioxidant assays like DPPH, where we did alpha amylase inhibitory assays for anti-diabetic properties. That was that was initial characterization. We also did phytochemical characterization.
Excuse me,
Yes.
Mr. Ali. I would like to say sorry. Our time is very important this on the symposium.
Okay. Yeah,
time is shortened. Last 5 minutes, okay?
Yeah, I'll take it.
Please.
Yes. Thank you, Dr.
Please.
Ibrahim. I'll take it. Yes.
Thank you for your kindly.
Yeah. So these are brief phytochemical analysis. We can see that total phenolic contents and total flavonoid contents, and the alpha amylase inhibition was almost 50%. Similarly, we could show that increasing concentration of aqueous extract inhibited the free radical generation. I'm coming to now glycation, where I can show that the browning and fructosamine content, the early glycation products were quite significantly reduced in the plant extract. This green line shows the control, the glycation system, and the red bars shows the treatment or extract along with the control system. So we could significantly show that the early glycation products and Maillard reactions are significantly inhibited. Then we measured the advanced glycation products and glycation-induced aggregation. Here also we showed that the inhibition was quite significant in the presence of Nigella sativa extract. We measured the total amount of advanced glycation product using spectrophlorimetric observations. We could show here also that Nigella sativa extract was quite efficient in reducing the total amount of ages. We did glyoxidation. We checked the effect of Nigella sativa extract on DNA damage, and we could show that Nigella sativa extracts were able to prevent the glycation mediated DNA damage. This work has been published in the first issue of CupMap. We have also worked on thymoquinone, the phytoconstituent of Nigella sativa extract, where we could show that thymoquinone was also very efficient in inhibiting the browning, fructosamine content, and carbonyl content. We could also show that aggregation was also significantly reduced by this Congo red and thioflavin T assays. The total ages was almost reduced by 78% by a higher concentration, that is 20 micromolar of thymoquinone. Then we did an fluorescent quenching study where we showed that there is an interaction between the BSA and thymoquinone that is very important when you are going to develop a drug. So we could show that increasing concentration of thymoquinone was more able to quench the absorbance of BSA. We did the similar kind of studies with the thymoquinone, which we had done with Nigella sativa, and we could show that thymoquinone was also very efficient in reversing the strand breakage of the DNA. So this some of this work has been published in one of the journals of natural volatiles and essential oils, where we have shown the anti-glycating and anti-aggregation potential of. So with all these experiments and results, we could establish that the Nigella sativa was quite efficient in inhibiting the production of early and advanced glycation products. It was also able to prevent the glycation-induced aggregation, which we checked with the aggregation of proteins. The black cumin seed extract was also able to prevent DNA damage, which indicates its role in the role in preventing the oxidative DNA damage may be a combination of anti-glycating and antioxidant potential of black cumin extracts. And our preliminary studies also suggested that alpha amylase was inhibited by extracts. One of the major significant findings was that thymoquinone was more potent as compared to the extract in preventing the formation of glycation products. So the conclusion of my study is that the aqueous extracts of Nigella sativa seeds clearly show anti-glycating, anti-aggregation, and this property may be because of antioxidant property also, because now people are suggesting that this antioxidants also can work as anti-glycating agents, because one of the mechanisms by which glycation works is to generate the free radicals. So these seeds maybe can be further characterized and can be used for significantly preventing the secondary complications of diabetes like glucotoxicity, can be used for preventing the protein aggregation mediated neurological disorders, and pure compounds can be used to develop drugs on the basis of them. So this work has been mainly done by two of my students, Prerna Malik and Dinesh Kumar. I have received fundings from various funding agencies, and my collaborators are this. Thank you. for giving me this opportunity. And if any question, please.
Thank you, Mr. Ali. Should have any question? No. Okay. Please, Mr. Şekeroğlu.
Do you hear me? Have you hear me?
Yes. Yes,
Mr.
I can.
Ali,
Yes.
thank you
Yes.
for your nice presentation. You know, our Prophet Hazreti Muhammad said that Nativa Sativa, Nigella Sativa have cure all disease. You should
Yes.
use the seeds mostly care. You know?
Yes. It
Yes.
it cures for everything but death.
Okay.
Yeah,
Okay.
Yes.
Thank you for
Yes,
your.
thank you.
Diğer sunuma geçebiliriz. Kalbak rahmet.
Thank you, Professor.
OP3, OP3. Sevgi hocam.
Yes.
OP3 sunabilirsiniz. Davet edebilirsiniz.
Okay.
Kalbak rahmet.
OP3. Huh? OP3. Yes. Next presentation. Kalbaza Ahmet Yasin. The name effect of Lawsonia inermis ointment on excisional wound healing in Vista rats. Yes, Yasin, are you ready?
Yeah, I'm ready. Good morning. Can you hear me?
Yes.
Yeah, good. So good morning, everybody. Before starting, I would like to address my thanks to the organizers of this symposium, especially Professor Nazim, to you, Professor Sevgi Kolayli and Professor Ibrahim Tumen, the chairs of this session, and to all the other participants. I'm very pleased to be with you today, and I hope we will meet together in better conditions soon. So I will first share my presentation. Does my presentation appear on the screen?
Yes, it's okay.
So I will start. So allow me please today to present my study entitled The Effect of Lawsonia inermis on Excisional Wound Healing in Oyster Rats. We will start with an introduction. Wounds are frequently encountered in humans and animals. After a wound, an intricate process takes place into 4 overlapping phases, which are hemostasis, inflammation, proliferation, and maturation. This process can, however, be delayed by several factors, which can result in serious consequences and lead to amputation in complicated cases. Therefore, the search for new agents that can improve the healing process is of great importance, and many efforts have been made in this field. The use of traditional medicinal plants for wound healing have been widely known by ancient populations. It is based on their antiseptic, astringent, anti-inflammatory, and antibacterial properties. Lawsonia inermis, commonly known as henna, is a member of the family Lythraceae, mostly spread in tropical regions. This plant is widely used by people due to its antimicrobial and Is there a problem?
No problem.
No.
Okay.
No.
So I can continue, sir.
Yes.
So the plant is widely used by people due to its antimicrobial and astringent activities. However, its main use is cosmetic as a dye for hair and skin. The aim of this study was then to assess the healing effect of Lawsonia inermis or henna leaves applied in the form of an ointment on excisional wounds in Western rats. as material and methods. So the study was carried out during the period from October to December of the year 2019 at the Institute of Veterinary and Agronomic Sciences of Betnawan University in Algeria. For the realization of this study, 20 healthy Western rats were obtained from the Pasteur Institute of Algeria. Before starting the experiment, animals have been adapted to the new regimen for 10 days in a well-ventilated and temperature-controlled room. They have been maintained in clean rodent cages where they have received standard rat pellet, feed, and ad libitum water. After the creation of wounds, rats have been randomly divided into two groups of 10 each. The first group or control group have received a daily application of petroleum jelly as a treatment for his wounds, while the second group have been treated with an ointment prepared from the plant Lawsonia inermis. Evaluation of wound healing was performed macroscopically by measuring wound surfaces, calculating wound closure and closure percentage, and by determination the epithelialization period during 23 days. After that, a histological study of the formed tissue was performed, and finally, results obtained in both macroscopic and histological studies have been subjected to statistical analysis. To prepare the ointment, Lawsonia inermis leaves have been gathered from the region of Tiart in West Algeria. After complete drying of the leaves at an optimal temperature, they were crushed by an electric grinder until a fine powder was obtained. An ointment formulation of 50% was then prepared by adding the leaves powder to petroleum jelly, heated until being melted in 65 °C water bath. The ointment formulation was then mixed until a homogeneous mixture was obtained and finally stocked in a glass bottle. So before starting the wound creation, we have respected all aseptic conditions by sterilizing the instrument. After that, the animals have been anesthetized with an intramuscular injection of ketamine and xylazine. The dorsal part of the neck was shaved and the skin to be excised was outlined with a marker. We have not used any antiseptic before surgery. A rectangular excisional wound of approximately 700 mm square was then made along the margin. And after wound creation, hemostasis was achieved by the application of a sterile gauze soaked in normal saline. On the next day, or at the end of the surgical procedures, animals have been maintained in their cages with their wound left open. On the next day, we have started the treatment of the excisional wounds as already described in the experimental design. In the macroscopic study, the progressive changes in the wound were monitored on an HD camera each 3 days. Wound pictures have been uploaded on AutoCAD to measure the wound area surface. The measures obtained have been used to calculate the wound closure percentage using the following formula. Percentage of wound closure equals wound surface at day 0 minus wound surface at day n, split by wound surface at day 0, multiplied by 100, where n equals the day 3, 6, 9, 12, 15, 18, 21, and 24 of the study. Finally, and to determine the endpoints of healing, wounds have been observed daily to detect the moment of scab dropping without leaving any wound growth. At the end of this macroscopic study, animals have been sacrificed by an intracardiac injection of propofol. The tissues were excised from the wound site of each animal and have been separately stored in 10% formalin solution. Samples underwent the habitual histological procedure to prepare sections, which were subjected to a microscopic evaluation to assess epithelialization, differentiation, amount of granulation tissue, degree of inflammation, collagen fiber orientation, and neovascularization. The microscopic observations were then transformed into scores according to a modified scoring system based on the previous model adapted by Sultana et al. And Abramov et al. As you can see in this table, we have given scores from 1 to 3 to each parameter to the study, and finally the total histological score was calculated. So the obtained results in the macroscopic and histological studies have been treated statistically using the 19th version of Minitab on Windows. The test student has been used to compare the two groups of the study and determine which one has the better healing. As a result, now, our macroscopic results have shown that the group treated with Lawsonia inermis ointment had better healing than control group during all the experiment. And the difference between both groups is considered as highly significant, as you see in the table. Moreover, rates treated with the plant ointment have completed their epithelialization in a shorter period compared to the rest of the control group, who have shown covered wound with a scab even at the 24th day of the experiment. These results are better illustrated in the present figure, but shows the macroscopic appearances of wounds in both groups. And as you can see, healing was always better in Lawsonia inermis group. The obtained results in the macroscopic study have been confirmed by the histological results, showing that Lawsonia inermis treated rat had the best total histological score. This was mainly due to the enhancement observed in terms of epithelialization, inflammation, collagen fiber orientation, and angiogenesis, where the Lawsonia inermis group had a significantly higher score than control group. However, even if the recorded scores of the differentiation and granulation tissue were higher in the treated group, we have not perceived any significant difference in comparison with control group. The histological sections realized after 24 days have shown in control group the presence of a scab, a large amount of granulation tissue, few collagen fibers, and few neovessels. However, in Lawsonia inermis group, a net epithelialization is present with a differentiation, many neovessels, and a small amount of granulation tissue. This result can be explained by an enhancement of the different wound healing phases, especially the inflammation process with a possible antibacterial and anti-inflammatory effect of the plant, the proliferation phase by a possible stimulation of fibroplasty and thus an augmentation of the collagen synthesis, a stimulation of epithelialization and angiogenesis, and finally the plant has enhanced the maturation phase, possibly by enhancing the collagen maturation and the transformation of fibroblasts into myofibroblasts with contractive properties. Even if this study has proved the good healing effect of Lawsonia inermis in excisional wound model, we cannot yet relate this effect to a specific component. However, the studies of the chemical composition of this plant have reported its richness in coumarins, known with their antioxidative properties, in flavonoids that are good antibacterial and astringent agents that prevent cell necrosis, stimulate angiogenesis, inhibit prostaglandin synthesis, and modulate cytokine expression. In tannins that are also good astringent and antibacterial, antioxidative and anti-inflammatory agents that stimulate fibroblast and keratinocyte proliferation and participate in the chelation of free radicals. And finally, in alkaloids that block the metabolic pathway of arachidonic acid men. We can conclude from this study that the topical application of an ointment prepared from the Lawsonia inermis leaves powder improved wound healing in an excisional model. The Lawsonia inermis ointment used in our study showed better results because the wound contraction and the epithelialization period were better than those observed in control group. This finding are asserted by the histological finding after 24 days, which shows an almost healed skin with good epithelialization and differentiation, as well as good angiogenesis. This study is already published in the journal Veterinary World and can be taken as a benchmark for further investigations, which will aim to isolate the various compounds of the plant and determine the exact effect on wound healing. And thank you for listening. I can't hear you, Professor.
Sevgi Hocam, sesinizi açın.
Thank you for your beautiful presentation, Dr. Kalbaza. Are you hear me?
Yeah, I hear you. Yeah, I do.
Yes. Do you study diabetic wound healing in this plant?
We did not yet study the diabetic healing, but it's in our perspective. So since it's a preliminary study, we have adopted a simple model, but in the future we will try to test the plant on diabetic animals and also on burns to enlarge its application.
I advise, please.
Yeah,
You should study. The results very good.
Maybe this year we will perform the study, and hopefully next year we will share the results with you.
Okay. Thank you very much.
Thank you.
And another presentation. Next.
OP4.
For influence of environmental factors on inhibitory activity against PCF1 gene expression and habitat suitability mapping of cordatum makino in Japan. And Donna Alsadi will presentation. Okay?
Yes.
Welcome.
Hello, everybody.
Alsadi.
Hello, everybody. My name is Doa, and I'm a researcher from Iraq.
Very good name.
Now, currently, I am a PhD student at Komoto University in Japan. And today, I would like to talk about my research. So the plant we're talking about is a Cardiocranium caudatum, also known as Obioli. So this plant is from family Liliaceae and it has English name giant lily. And as I mentioned, the Japanese name is Obioli. This plant habitat is very limited to Japan and some area in Russia. And the edible part of this plant is the leaf and the root. It also can be used as ornamental plant because of the scented flower, as you can see in the photo. So this plant has a lot of nutritional value and is used as a medicine and also as a edible food in Japan. But because of the additional developmental activity and sometimes even natural disaster like landslide, typhoon, and many other events, sometimes the zone where the plant grow usually disappear and many of these species also like vanish. So we try to make conservation effort for this plant and based on the soil analysis, study of the environmental data from the study area. And the study area we selected here is the Shibujima Oki Island in Japan. So our objective is to study the investigation of this plant in the Shibujima Island and also investigate the environmental data in this island and check if the inhibitory activity against BSF1 gene expression has any correlation with this environmental factor, and also making a habitat suitability mapping for this plant. So here is the study area in the Oki Island, as you can see from this map. And Shibujima Island is located in the southern part of Oki District, Shimane Prefecture in Japan. It's considered one of the smallest islands and with maximum elevation of 325 meter. And this island, like many other islands in Japan, was formed because of volcanic eruption and was covered with many mountains. And the temperature ranges between 35 degrees in summer to -3 in winter, with adequate amount of rainfall also. So we did the in situ soil analysis with the soil pH, volumetric soil moisture content, and soil bearing capacity. For you who don't know about soil bearing capacity, is like a parameter that measure the resistance of the soil to the pressure. So when the plant root try to penetrate the soil in order to reach the water resources, sometimes they get resistance. And if this resistance is high, that means the soil bearing capacity is also high. So it's not good when we have high value for this. Also, it's not very good to have very low value for this, because that means the soil is very sandy and it's difficult to keep enough amount of water for the plant growth. We also check the chemical analysis of the soil. We check the ammonium, nitrogen content, the phosphorus, potassium, magnesium, manganese, iron, copper, zinc, and boron content. We check pH and electrical conductivity also. We also examine the solar radiation, the effect of solar radiation on this plant. As you can see here, we stick optoleaf film on the leaf to measure the solar radiation and the discoloration rate, and finally also the what we also call the fading rate. We also measure the photosynthetically active radiation bar. This measurement is important because this plant usually prefer shady area for its growth. So when it's exposed to the sun, usually the leaf will undergo some discoloration and the color will be fade a little bit. So here we have a photo of two leaves, and the right side you can see the tip of the leaf a little bit have a discoloration of the green color, a little bit yellow color because of the exposure to the sunlight. And as you also saw from the photo, this plant has two different sizes for the leaves. Next is the gene expression, the BSF1 gene expression. This like a simple introduction first for this gene expression. So this is a subunit also from the complex called GINS. And this complex, actually the name came from Japanese, which means Go Ichi Ni San 5123, because it's composed of the SLD5, BSF1, BSF2, and BSF3. And it's very important for the DNA replication, and it's expressed in the many tissue, like in the bone marrow, thymus, testes, ovaries, and in many immature cell lineage, and also it can be seen in the embryogenesis, spermatogenesis, and the loss of this BSF1 lead to the embryonic lethality in the mice, and also delay the induction of hemopoietic stem cell proliferation. So in cancer stem cell, the complex has overexpression, all the component of this complex, especially BSF1 and SLD5 and the melanoma. And the high transcriptional activity of gene also examined in the edge and the edge and also vascular region of the tumor. So knocking down this BSF1 can lead actually to growth arrest. During the cell cycle, it can lead to the growth arrest by increasing the fraction of the cell in the G1, S, and G2 and M. This has more effect on the cancer cell than the normal cell. So in the method for this one, we isolate the BSF1 promoter from the mouse, and then stained with the enhanced green fluorescence protein, transfection later to the cell line. We have a Lewis lung carcinoma cell line. So we established the Lewis lung carcinoma cell line with the BSF1 that is stained with the EGFP. And by adding the plant extract, we examined the morphological changes and also the intensity of the staining with the EGFP, and later with the BI, and also the quantitative the PCR. This actually, the activity is not the first time discovered, because we have Nobuyoki Sensei is the co-author in this research, and he got a patent for his research on this plant and this inhibition activity for BSF1. So the aim of this study is not to check if this plant has BSF inhibition or not, because it's already established. We just want to see if this activity is related to the...
Sorry, time limited, please.
Oh, already? 5 minutes left. Sorry. So we prepare the data and for habitat suitability mapping. We use the Maxent model, and we use the topographical feature, soil condition, and meteorological feature also. And we consider also the accuracy of this final habitat mapping by using ROC. This is interpolation map from the soil and the geographical information, and also the meteorological information. And this is the final habitat map of the plant. By combining the interpolation map of all the parameter we have, we got the area which has a high suitability for this plant and has lower suitability, which can also use for conservation and future cultivation and preservation of this plant. This is also the validation, the final validation for the habitat suitability mapping. As you can see, is very high precise. And for the BSF1, we saw that the gene expression was affected by the soil parameter, specifically the iron and the phosphorus level. Like we saw that the plants that are growing in soil with high iron exhibited low inhibition for the BSF1, while the plant that are growing in the soil with high phosphorus level exhibited high inhibition for BSF1. So the chemical content of the plant was affected by the soil parameter. We're still investigating the HPLC and data on the elucidation and for the chemical content and which one is responsible for that. And we also established the suitability of the land for the plant for this plant growth. So thank you for listening. This is a good photo for my lab, but unfortunately we should have like more members, but we couldn't take a photo because of the Corona situation. Hopefully in the future we have like a group photo and also with everyone in this symposium in the future also. Thank you for listening.
Okay, thank you. Thank you very much for your interesting study also.
Thank you very much.
Wow, nice to meet you.
Thank you, nice to meet you too.
Okay, are there any questions? Everybody? I think no question.
Thank you very much.
Okay. And next presentation, OP, I think 5. Okay.
Okay, OP5.
OP5, yes.
Denisa Batır, Marin.
Biological assessment of alcoholic extracts, Equisatum species, and antioxidant and neuroprotective activities. Deniz Batır Marin will presentation. Okay.
Everyone, can you hear me?
Welcome, Deniz Batır.
Yes. Everything is okay? You can see the presentation?
Yes, yes. Go on, please.
First of all, I would like to thank Professor Shekeroglu and all the organizing committee for the opportunity they offered us by organizing this Congress in the condition of the pandemic we are in. My name is Denisa Batır Marin, and I'm going to present my work entitled The Biologic Assessment of Alcoholic Extracts of Ecvisetum pratense, Ecvisetum sylvaticum, and Ecvisetum telmatea, Antioxidant and Neuroprotective Activity. Just a moment. The Ecvisetum species are perennial plants which have spontaneously developed and are frequently found in forests, wet meadows, and swamps. Sterile stems belonging to the Ecvisetum genus are often used in traditional medicine. The purpose of this work is to highlight the chemical composition of the extracts obtained from the Ecvisetum species and to investigate the biological action in vitro and in vivo. In vitro evaluation of the antioxidant activity of the plant extract, methanolic and ethanolic, obtained from 3 species of Equisetum genus, was determined using 4 tests. The chelating capacity of ferrous ion, the lipoxygenase inhibition capacity, the scavenger action of the hydroxyl radical, the scavenger capacity of the superoxide anion. The extracts were obtained using methanolic and ethanolic alcohol in 70% concentration. For the in vivo, we determined the neuroprotective activity of those 3 extracts, only ethanolic samples, using zebrafish tests. The behavioral tests were made were the swimming pool test and the anxious behavior of the Y labyrinth test for short-term memory determination. We used only ethanolic extracts because the methanolic ones have shown to be toxic for zebrafish. Here are the results for the in vitro evaluation of the antioxidant activity of the plant extracts obtained from the Equisetum species. The first one, the chelating capacity of ferrous ions, is an important indicator for the evaluation of antioxidant activity. The results obtained in this test are shown in these 2 figures. The methanolic extract, 70% of Equisetum sylvaticum, is more active compared to the gallic acid. This time, the phytocomplex present in the extract achieving an additional synergy compared to the single compound. Sorry. The second one, here are the results obtained for the determination of the lipoxygenase inhibition capacity. Comparing for each plant species analyzed, the 2 types of extracts, it is observed that the methanol extracts are more active and than the ethanol ones for Equisetum pratense. But for Equisetum sylvaticum, it shows the best lipoxygenase inhibition capacity. The scavenger, the results obtained when evaluating the scavenger capacity of the hydroxyl radical are presented in these 2 pictures. The methanol extracts of the species Equisetum sylvaticum showed the most important action, being slightly higher than the reference substance, gallic acid. The lowest scavenger activity of the hydroxyl radical was recorded to the hydroalcoholic extract of Equisetum pratense. The last one, the scavenger capacity of the superoxide radical anion, is presented here. It is observed that the antioxidant efficacy for the test is closer as a value for the methanol extract of the Equisetum sylvaticum and Equisetum telmatea as compared to the reference substances, the gallic acid. As with all other determinations, the antioxidant activity for Equisetum pratense showed the lowest activity. Here are the results for the in vivo evaluation of the neuroprotective activity of the plant extracts from Equisetum species. The behavioral tests carried out followed the effects of the ethanol extract of this species of Equisetum on the anxious behavior, the swimming pool test, and short-term memory, the Y maze test. The behavior observed during the swimming pool test is used to assess the level of anxiety and desire to explore. An anxiolytic agent increased the time spent in the upper part of the tank, as well as the number of entries. For each type of Equisetum extract, individual tests were performed at both 0.5 milligram per liter and 1 milligram per liter ethanol extract. The ethanol extract from these 3 species of Equisetum have positive effects in assessing anxious behavior. Zebrafish exposed to concentration of 1 milligram per liter ethanol extract of Epizetum pratense and Epizetum sylvaticum exhibit anxiolytic and antidepressant effects higher than the concentration of 0.5 milligram per liter. For the ethanol extract from Epizetum termatea, it can be observed that the time spent in the upper area of the basin is higher of the in the concentration of 0.5 milligram per liter compared to 1 milligram per liter. Compared to the reference substance, the imipramine, the number of entries in the upper area of the basin for the zebrafish was higher at the concentration of 1 milligram per liter, while the concentration of 0.5, the number of entries were lower but comparable with it. The next test was the Y-maze test. Is based on the tendency of zebrafish to explore a new environment they are in. The percentage of spontaneous alteration is defined as the ratio of the number of alternations performed and the number of possible alternation. This behavior reflects spatial working memory, which is a form of short-term memory. For each type of Epizetum extract, individual tests were performed at both 0.5 and 1 milligram per liter ethanol extract. The ethanol extract from this species of Epizetum had positive effects improving short-term memory. The difference between the ethanol concentration of 0.5 milligram per liter and 1 milligram per liter were not significant, both with the role of improving memory performance in a dose-dependent manner. Compared to the reference substance, the donazepam, both the 0.5 and 1 milligram per liter concentration in these three species caused an even twofold stimulation of short-term memory by increasing the contribution of spontaneous alternation. The obtained results indicate that methanolic and ethanolic extracts of Epizetum sylvaticum have the greatest antioxidant capacity for all 4 in vitro tests made, followed by the ethanolic extract of Epizetum termatea and Epizetum pratense. The results for the in vivo test regarding the effort of short-term memory, significant differences are observed and confirm the great effectiveness of the Epizetum pratense and Epizetum termatea extracts compared to Epizetum sylvaticum. Also, the 1 milligram per liter concentration of Epizetum pratense ethanolic extract had anxiolytic and antidepressant effect higher than the other 2 extracts used. Thank you very much. And I'm sorry that I was very emotional. Any questions?
Yes. Thank you, Deniz Batır, for a nice presentation. And I want to ask some short question. Why are you not used FRAP test, ferric reduced antioxidant power? Do you know?
We also used the DPPH test, but I didn't put it here. The results were the same. Epizetum sylvaticum had the best antioxidant activity.
Yes, but FRAP, ferric reduced antioxidant test, is total antioxidant capacity test and is very basic and easy. I advise in other studies.
Yes,
maybe
for sure.
You can use.
Sure,
okay,
yes.
Thank you very much.
Thank you.
Is there any questions? I think no question. And our section, last presentation, Asma Shahbaz, pre-formulation of pre-formulation study for nano-emulsion preparation of essential oils for treatment of candidiasis. Okay, are you ready?
Yes, I am ready. Thank you so much.
Welcome.
Thank you.
Okay, you can go on.
Yes. Are my slides visible?
Full page. Okay.
Good morning, ladies and gentlemen.
Good morning.
Asma Shahbaz. I'm a master graduate student from Near East University, Turkish Republic of Northern Cyprus. My topic for presentation is pre-formulation study for the nano-emulsion preparations of essential oils for the treatment of candidiasis. My synopsis of my study will include the explanation of candidiasis, essential oil, and the treatment strategy using nano-emulsions. And by the end of the presentation, I will conclude my study. Talking about candidiasis, candidiasis is a disease caused, or the source of this disease is a fungi, which is a yeast-like fungi that causes candidiasis, Candida albicans. It's found as normal flora in human body as well, but an overgrowth of this Candida at a certain magnitude that it causes inflammation action will lead to a disease which needs to be cured. And it's most likely to be found on skin and nails. Candidiasis is one of the very common diseases for the skin and the nail infections, causing nail infections as well. Moving towards essential oils, as we all know, essential oils are the natural aromatic compounds that are obtained from the plant source. Any part of the plant can be used to extract the essential oils. It can be from the bud, leaves, flower, bark of the plant. They are the complex mixtures of terpenes and terpenoids. Terpenes are the natural hydrocarbons, while terpenoids are related to terpenes, but they can differ from the rearrangements of the oxygen group on it, on the structure of it. It can be acidic, alcoholic, aldehyde, can be an ester or ketone as well. They are the organic solvents, and their metabolites can be volatile or non-volatile. The essential oils that we used in my study are cinnamon essential oil and the clove essential oil. Cinnamon oil is obtained from the bark of the plant, while clove oil was obtained from the bud, the flower bud of the plant. Talking about nanoemulsions, nanoemulsions are kinetically stable biophysics dispersions of two immiscible liquids, oil and water. Since they are immiscible to each other, they tend to increase the interfacial tension, which this interfacial tension has to be stabilized by adding a stabilizer, a surfactant, or an emulsifier. And nanoemulsions are can be of two types: oil and water, water and oil, or it can be double emulsion with the double layers of oil and water, and water and oil, water and oil. They are advanced nanodroplet system for the systemic controlled and targeted drug delivery system. My study for the essential oil is in two steps. First, we analyze the constituents found in our oils, and then the assays were performed to check their antifungal activity against Candida species. The assays we performed for the antifungal activity was the broth microdilution test and disk diffusion method. The broth microdilution was done to check the minimum inhibitory concentration of the oil that can be enough to inhibit the growth of Candida, and the disk diffusion test again for the inhibition of the Candida species. Cinnamon oils, source Cinnamomum xylanicum from the family Laurier, clove oil Eugenia caryophylleta, two oils used. And their constituents were analyzed by using a technique called gas chromatography for elution of the compounds or the components found inside the essential oil. And the results yield that cinnamon oil is consisting of an aldehyde as the major constituent, or the abundant constituent that was as high as 61.5%. And in the clove oil, we found an alcohol, eugenol, which was found in abundance, 79.3%. Moving towards the assays, the first test performed, this diffusion method, was to check the activity of the inhibition activity of our oil against the Candida species. And the result shows that our cinnamon oil, here we can see that the diameter zone of inhibition by the cinnamon essential oil was way higher, wider as compared to the clove oil. Both of them are active against the inhibition, but the cinnamon oil shows better results when compared with the clove oil. And broth microdilution test was performed on both oils again, and the comparison was done. And we can see that the cinnamon essential oil was active and at 0.001 microgram per ml, while clove oil at 0.008 microgram per ml. Here again, we can see that the cinnamon essential oil was strongly active against the inhibition of the candida when compared to the clove oil. So depending on those results, a nano-emulsion pre-formulation was prepared using non-ionic surfactants, and cinnamon oil was used as our active and as well as our oil phase in our nano-emulsion system. And purified water was used as an aqueous phase, and we obtained an optimum formulation. This optimum formulation was nano-sized droplets. It was a nano-emulsion, and this optimum formulation was then subjected to the characteristic study of the nanoemulsions like droplet size, the polydispersity index, and the zeta potential. And the results were quite optimized, and there were good results were obtained. And I would conclude my study that the cinnamon essential oil, when compared with the clove essential oil, the test results shows that cinnamon oil is way more active against this inhibition of the Candida albicans when compared to the clove oil, and it has an effectivity against the treatment for the candidiasis if formulated in a suitable dosage form. Since the formulation we prepared was a pre-formulation, further study needs to be done in this, and it can be processed more to obtain a formulation in the end. And of course, a dose will be an adjustable dose should be maintained for this in order to make it active for the use in the treatment of candidiasis. And yeah, thank you so much. I would like to acknowledge my professor, my advisor, for Dr. Yildiz Azal, for her immense support and guidance, and my all team members, my friends, for their cooperation during this study throughout this pandemic, which was a really tough situation and time for all of us. But we made through it, and we really worked hard for it. I really thank them all, and of course, my special thanks to the organizers of this symposium. Thanks a lot for making this platform possible for us to share our knowledge and gain information and knowledge from this symposium. Thank you so much. I would like to open for the questions if anybody has some questions to ask, please.
Thank you. Also, Esma Shahbaz. Are there any questions for your nice presentation?
Thank you.
Say all salams to Cyprus also. Is there any question? No question, I think. We have completed this first section today, and thank you again to all participants. Okay.
I would like to thank you, the chairs of this session and the speakers. It was very fruitful and perfect presentation. We are very happy to entertain you at MESMAP. And I kindly close this session, and we will meet you the second session at 11 o'clock Turkish time, Istanbul time. Please be ready before 11 o'clock.
Okay.
Thank you again.
Okay. Thank you. Thank you.